The overall goal of the following experiment is to image and probe the physical properties of phase-separating membranes using atomic force microscopy. This is achieved by first mixing dioleo-phosphatidylcholine, sphingomyelin, and cholesterol at a 2:2:1 ratio to form a phase-separating lipid mixture. Next, the supported lipid bilayers are prepared by depositing the liposome suspension into a mica surface. Using atomic force microscopy, the surface of the bilayer is then scanned, producing a topographic image where structural features like height can be measured. Next, force spectroscopy is performed on specific areas of the bilayer to probe the physical properties of the bilayer, most notably the breakthrough force and membrane thickness. Ultimately, the results show differences in properties between different lipid phases based on AFM images and force curves.
The main advantage of this technique over existing methods, like fluorescence microscopy, is the increase in resolution and additional properties AFM can probe on the sample.
This method can help answer key questions in the field of biophysics, such as the structure of biological membranes and proteins embedded on such membranes. Furthermore, we can also look at the physical properties of the membrane and monitor how changes in the composition or addition of membrane active agents might change these properties.
Generally, individuals new to this method will struggle because of the fragile nature of the sample. This has implications on sample preparation, handling, choice of cantilevers, and AFM imaging and spectroscopy conditions.